Welcome, curious minds, to the deep dive. Today, we're diving headfirst into a world that's, well, not just evolving, it's truly revolutionizing healthcare as we know it.
Absolutely. We're talking digital health technologies.
Right. Those incredible wearables maybe on your wrist right now, the smart devices fitting into your daily life, and these advanced sensors,
Yeah.
they're all quietly collecting just this huge stream of real-world health data.
And they're not just gadgets, are they? They're really becoming the sort of central nervous system for modern clinical trials.
Yeah, offering insights into our well-being that, frankly, were unimaginable not long ago. Picture this. Your doctor understanding your health not just from, that single yearly checkup.
That snapshot in time.
Exactly. But from continuous dynamic monitoring. It paints a full, personalized, real-time movie of your unique health journey. It's incredibly exciting stuff.
It really is the sheer amount of data, the detail. It's immense.
And how granular it is.
Right. These technologies take us way beyond those occasional clinic visits. They give us this ongoing, almost intimate picture of a person's health, out of the real world, not just in a controlled lab setting.
It's like moving from a few photos to a whole documentary of someone's physiological story.
That's a great way to put it. And that level of insight, it holds massive promise
Yeah.
for understanding diseases, developing treatments, ultimately for personalizing healthcare in ways we're just starting to grasp.
That's the real promise, isn't it? That shift from a fragmented view to this continuous story.
Mhm.
But here's where it gets, well, really interesting, and where we find this hidden but absolutely critical challenge, this huge challenge that kind of underpins this whole revolution.
You mean the data itself?
Exactly. All this incredible high-frequency data collected minute by minute, day by day, it comes with an often overlooked problem: missing data.
Ah, yes. The gaps.
And we're not talking about just a minor annoyance here. We're talking about inevitable, sometimes systematic gaps in these continuous streams.
That really is the crux of it. These gaps, they might seem small, but if you don't handle them with extreme care, well, they become a major statistical problem.
How so? What's the real danger?
They introduce bias, which means the data we do have might not accurately represent the whole picture.
So the remaining data is kind of skewed.
Precisely.
Yeah.
And the downstream effect.
Yeah.
This bias can actually invalidate crucial clinical trial endpoints.
Endpoints. Those are the things they measure to see if a treatment works, right? Like, did symptoms improve?
Exactly. Those measurable outcomes improve mobility, reduce pain, whatever the trial is looking at. If those endpoints are compromised because of biased missing data,
Then the whole trial's conclusions are shaky.
fundamentally questionable, yes.
Yeah.
And that affects whether a new therapy can even reach patients.
So what does this mean for you, our curious listener, and for the future of medicine that relies so much on this data? How do we make sure the data is reliable, trustworthy, when bits are just missing?
That's the big question.
And that's our mission for this deep dive. We've put this exploration together just for you to really unpack this complex, often hidden world of missing data in digital health.
We'll look at why the old ways, the traditional statistics, just don't cut it anymore.
Right. We'll dig into the unique challenges these high-frequency digital health data sets throw at us. Some are pretty counterintuitive.
And then the exciting part, the innovative solutions being developed.
Yeah.
Really clever stuff from data scientists and statisticians.
And finally, we'll understand what this all means, the implications for healthcare, for clinical research, and ultimately for the treatments that might one day help you.
And our main guide for this discussion is a really insightful paper. It's titled Considerations to Address Missing Data when Deriving Clinical Trial Endpoints from Digital Health Technologies.
That's comprehensive.
It is. It's by a team of experts, Junrui D, Charmaine de Manuel, and others.
Yeah.
They really lay out the problem landscape and, crucially, the path forward. It's like a roadmap for ensuring rigorous science in our digital health future.
Okay, let's get into it. Let's unpack this invisible threat. Why is missing data, which sounds maybe kind of minor, actually such a huge deal in digital health?
Well, first, we need to really appreciate just how transformative these digital health technologies, or DHTs, are in clinical trials. It's not overstating it to say they're becoming the backbone of modern medical research.
A whole new paradigm, really.
Absolutely.
Yeah.
Think about the old way.
Yeah.
A participant goes to a clinic maybe once a month, every few months.
Right. For tests, measurements, questionnaires.
Exactly. And you get these specific isolated data points,
Yeah.
like snapshots in time.
A few still photos.
Right. But now, with DHTs Smartwatches, specialized sensors, even smartphone apps were collecting data continuously.
Hmm.
Physiological data, behavioral data.
All the time.
Pretty much.
Yeah.
And that just fundamentally changes what we can learn about a patient.
It moves beyond those, maybe slightly artificial clinic visits.
Hmm.
These technologies let researchers capture a much more holistic, nuanced, more accurate picture of someone's health journey outside the clinic, in their real life.
We can track sleep patterns minute by minute, activity levels all day, subtle changes in heart rate variability, even capture when symptoms flare up in real time.
It's not just more data, it's better data in a way, isn't it? Data that reflects real life.
That's the key, ecologically valid data. It shows how health and disease actually play out day to day.
And the promise there is huge. More data, higher frequency. You'd expect deeper insights, right?
Logically, yes. The potential to spot treatment effects earlier, to understand exactly how an intervention impacts daily life with, like, unprecedented clarity.
Finding patterns we just couldn't see before with the sparse data.
Precisely. Building this high-resolution, dynamic picture of health. Moving beyond simple averages to see individual journeys.
But, and there's always a but, isn't there?
Usually. With great data comes great challenges.
And alongside all that amazing potential is this inevitable, sometimes quite damaging problem of missing data. And you said it's more than just an empty space in a file.
Oh, far more. When we talk about missingness in DHT data, it's not just a blank cell. It's a fundamental statistical issue that can really undermine a whole trial.
How? What's the mechanism?
The main concern is statistical bias. It means the data points we do have might not accurately represent the underlying reality.
Because the missing bits aren't just random absences.
Often, no. Their absence itself can carry information, meaning what's left is skewed.
So it's a distorted picture we're looking at, potentially leading to wrong conclusions. And that distortion, that bias, that hits the clinical trial endpoints.
Exactly. If the bias is bad enough, it can completely invalidate those endpoints.
And those endpoints are everything, right? They're the proof.
They are the absolute bedrock. The measurable outcomes, reduced symptoms, increased activity that tell us if a treatment works, if a drug is effective, if a therapy improves quality of life.
So if those are invalidated because of dodgy missing data,
The entire trial's conclusions are compromised. It's not just a technicality. It means the evidence used to approve a new therapy could be fundamentally flawed.
wow, that's a huge concern for everyone. For you listening who might rely on these treatments someday, you want to know, absolutely, that the tech and the therapies based on its data are rigorously proven.
The stakes are incredibly high. Public trust, public health, it's all tied in.
Absolutely. Which brings us to a really key point you touched on earlier, why the old statistical methods, the ones we've used for ages in research, just aren't up to scratch anymore.
Right. The paper by Demar, DeManuel, and colleagues makes this very clear. Methods like last observation carried forward LOCF,
Yeah, I've heard of that.
or just simple mean imputation filling gaps with the average. They were designed for much sparser data, often cross-sectional data, not this continuous flood.
They just weren't built for the digital age, for these high-frequency streams from DHTs.
Exactly. There's a huge gap, really, between those old methods and this new type of data.
They assumed a certain randomness or stability in the missing data.
Often, yes. Assumptions that just don't hold true for this dynamic, continuous data flow. Think about tracking activity levels. If your device cuts out at noon,
LSCF would just assume you stayed at that noon activity level all afternoon, even if you went to sleep.
right, or maybe took the device off because you felt awful and couldn't move, it's like trying to judge a whole day based on one photo from lunchtime. Completely unrealistic,
Or, imputation, just plugging in the average activity for everyone in the study.
which totally misses your individual pattern,
Yeah.
the time of day effects, everything that makes the data rich. The authors state these methods are not easily applied to continuous digital data sets.
Because they just flatten everything out, they lose the detail.
They can't capture that rich, time-dependent structure. Applying simplistic methods to complex data, it leads to wildly inaccurate conclusions.
Which really highlights the need for brand new approaches, more sophisticated ways to handle this.
Definitely. Which leads us into the really unique challenges of missingness, specifically in DHT data. It's not straightforward.
You called it a labyrinth earlier, especially with this high-frequency time series complexity.
Yes, because DHTs capture data at these incredibly tiny epoch levels. Seconds, minutes.
So like a heart rate reading every 10 seconds for steps per minute. That's an epoch.
Exactly, that raw, granular data. But then, for analysis, it often gets rolled up, aggregated into daily or weekly summaries.
Like average daily heart rate or total steps today.
Right. And here's the tricky part. Missingness can happen at any of those levels.
Ah, so it's not just one problem.
Not at all. You could lose individual epochs, a single missed heartbeat reading, maybe, from a sensor blip.
Or a few minutes if the watch loses connection.
Yes. Or you could lose a whole hour if the fit was bad or it was charging. Or even bigger gaps, whole days missing.
If the person just forgot to wear it or decided not to.
Exactly. So you have missing epochs, missing chunks of time, missing entire days. Each level presents a different analytical puzzle. Needs a different kind of solution. It's layered.
That really drives home the complexity. It's not one size fits all. But maybe even trickier, you mentioned biased missingness and MAR not missing at random. That sounds concerning.
It is. It's probably the most dangerous type of missingness. NMAR is insidious because the reason the data is missing is directly tied to the value that would have been recorded.
Okay, can you give an example?
The source paper gives a classic one. Participants might take off their devices when they feel particularly unwell, or, crucially, when their symptoms are really bad, or maybe when the treatment they're testing isn't working well for them at that moment.
Right. So say I'm in that chronic pain trial. If I take off my tracker when my pain is unbearable,
Then the data the device does collect for that day will show an artificially low average pain level because the worst moments are simply missing from the record.
they're not captured. Wow.
It's not random, it's systematic. If patients consistently do this when pain is high or when the drug isn't kicking in, the data makes the condition look less severe or the treatment look more effective than it really is.
That's incredibly misleading.
The paper explicitly warns this can lead to overestimation of endpoint performance. And that's critical for you listening if a new drug seems, say, 20% more effective than reality, just because people ditch the device when it fails them. Well, that's a flawed basis for approval.
It really undermines the whole process. It could lead to approving drugs that aren't as good as claimed, or maybe missing safety signals because people experiencing side effects stop wearing the device.
Exactly. And compounding this, the paper points out a common but overly simplistic current practice, threshold-based exclusion.
What's that?
It's where researchers just decide to drop entire days of data from the analysis if the device wasn't worn for, say, a minimum number of hours that day. Like, if you didn't wear your tracker for at least 10 hours, that whole day's data gets tossed out.
On the surface, that sounds kind of logical. If you don't have enough data, maybe don't use the day. Keep the data set clean.
It seems straightforward, but it's risky. It can introduce huge bias. Think about it. You're essentially only analyzing the good compliance days.
You're throwing out days when maybe the person was less active or felt unwell or the device malfunctioned.
Exactly. You systematically skew the average towards higher activity or better health states. The source is clear. This practice reduces the validity of derived endpoints. The conclusions become less reliable, potentially dangerously optimistic.
So trying to clean the data this way actually makes it less representative of reality. You're throwing out important pieces of the puzzle just because they're not perfect.
Precisely. Which really underscores why we need these more sophisticated, nuanced statistical approaches. Which brings us to the exciting part, the cutting-edge solutions.
Okay, let's get into those. How do we actually handle these gaps intelligently, especially at the really granular epoch level?
Right, so one key area is imputing those tiny gaps at the epoch level, trying to fill in those missing seconds or minutes very carefully.
Because you want to preserve those fine-grained patterns.
Exactly, you need precision. And a powerful technique here is within-patient imputation.
Within-patient, meaning you use the person's own data?
Yes. It cleverly uses that participant's historical data to fill their own gaps. For example, if you usually have a high step counter on lunchtime, but the device cut out for an hour during lunch today.
It wouldn't just put 0 or the steady average.
No, it might use your average step count from previous lunchtimes to fill that specific gap. It respects your individual patterns, your unique rhythm.
That makes so much more sense. You're preserving the individual's health story.
Absolutely. And building on that idea, there's functional data analysis, or FDA. This is even more sophisticated.
Functional data analysis. How does that work?
Instead of seeing data as just a series of separate points, FDA treats each person's data over time, like their daily heart rate or activity, as a continuous function, a smooth curve. like a personal health waveform.
A waveform. So it sees the overall shape, the flow, the rhythm of the data, not just dots on a graph.
Precisely. It looks at your daily activity pattern not as points, but as a smooth, possibly complex curve unique to you when there's a gap.
It doesn't just draw a straight line.
No, FDA imputes the gap by fitting a smooth, statistically derived curve through the existing data around it. It preserves that inherent time structure, your individual rhythm.
So it avoids those weird, sharp jumps you might get with simpler methods.
Exactly. It gives a much more realistic, biologically plausible estimate. It's like smoothly sketching in the missing part of that waveform, making it look natural.
That sounds incredibly powerful for keeping the continuous data, well, continuous and accurate. But what about bigger gaps, like when a whole day's summary is missing average steps, time active?
Right. That's tackling missingness in the aggregated summaries. Here, we often turn to methods like multiple imputation or MI and other model-based approaches.
Multiple imputation sounds like it does the job more than once. Why do that?
It's clever. Instead of just giving one single best guess for the missing value, MI generates multiple plausible imputed datasets, maybe 5, 10, or even more.
Each one's slightly different.
Yes, each reflecting the statistical uncertainty around that missing value. You then analyze each dataset and combine the results.
Why is that better than just one guess?
It gives a more honest picture. It accounts for the uncertainty that comes with imputation. And crucially, MI methods leverage correlations. They don't guess blindly.
Meaning they use other information you do have.
Exactly. They look at everything available to make a highly educated prediction for that missing daily summary.
Like what kind of information?
It could be the participants' baseline characteristics, age, weight, health conditions. It definitely uses their data from previous days or weeks. Their own history is often a good predictor.
Okay.
And, really importantly, it can use data from other endpoints collected at the same time. If average daily steps are missing, but you do have their heart rate data, sleep data, maybe even medication logs for that day.
You can use those related pieces to inform the guess about the missing steps.
Precisely. Especially if, for instance, that better sleep correlates with more activity for that person. It creates a much richer context-aware estimate.
That feels so much more robust than just plugging in an average. It's using the whole tapestry of data you have for that person.
It is. But we can go even further. For really complex, messy, irregular time series data, we're now seeing the power of deep learning.
Wow. Deep learning for missing data in trials, like AI.
Essentially, yes. Specifically, models like recurrent neural networks, or RNNs, and especially LSTM's long short-term memory networks.
LSTMs, what makes them so good at this?
They're designed to model sequential data, like time series. And they have this ability to remember patterns and dependencies over long periods within that sequence. They can learn really intricate missingness patterns, even if they're nonlinear or influenced by hidden factors.
So they're not just looking at the data right before and after the gap. They get the whole history, the underlying rhythm.
Exactly. Think of it like an expert art restorer. Old methods might patch a damaged painting awkwardly. Deep learning, especially LSTMs, is like an AI restorer that studied thousands of that artist's works.
Understands their style, their patterns.
Yes. And can reconstruct the missing sections almost seamlessly, matching the original style perfectly. LSTMs can infer your likely activity based on your usual weekly rhythm, your sleep, maybe even how those change depending on other factors.
So they can rebuild the missing data with much higher accuracy, especially if the data is really variable or complex.
That's the promise. More accuracy, more biological plausibility than even traditional MI in those really challenging cases.
That's where AI directly boosts the integrity of trial data, potentially leading to more precise findings, faster approvals.
The potential is huge.
Yeah.
But even with these amazing statistical tools, the best strategy is still prevention.
Right. An ounce of prevention
is worth a pound of cure, especially with data. So proactive strategies to minimize missing data right from the start are absolutely crucial.
Makes sense. Where do you begin with prevention in a trial?
It starts with the basics, optimizing how devices are deployed and really focusing on participant training. Good data relies on participants being able and, importantly, willing to provide it consistently.
So clear instructions are key.
Absolutely. Thorough, user-friendly training on how to use the device correctly, wear it consistently, handle minor issues, and crucially, understanding why their data is so important for the trial, for future patients, empowering them.
I can see how just handing someone a device without that could lead to problems, confusion, frustration. They might just stop using it.
Exactly. And beyond the initial training, Using technology for ongoing help. Automated reminders.
You get a notification. Looks like you forgot your device today?
Something like that, yes. Gentle nudges can significantly boost compliance. And another really smart strategy is using run-in periods at the start of the trial.
A run-in period? What's that?
It's an initial phase, maybe a week or two, before the main trial data collection officially begins.
It's like a practice round.
Sort of. It helps participants get used to the device, integrate it into their routine,
Right.
without the pressure of it being official data yet. But critically, it's also a screening tool.
How so?
Researchers monitor compliance during this run-in. If someone consistently forgets the device or really struggles with it, maybe they aren't the best fit for the demands of the sole trial.
Ah, so you can identify potential high missing data participants early on.
Exactly. It helps filter for a more compliant group, reducing the chance of big data gaps later.
Yeah,
It's like a pilot phase for device use before the real data collection starts.
smart. It ensures a better quality participant pool. And another strategy you mentioned is designing studies with patients. Co-design.
Yes, this is a huge shift. Instead of researchers just designing everything in isolation, co-design means actively involving patients, patient advocates, in the process.
In designing the study itself
Yeah.
and choosing the tech.
Yes, getting their input on the protocols, the devices, it enhances the real-world usability. Make sure things actually work for people in their daily lives, aren't too burdensome or awkward.
That makes so much sense. If a device is uncomfortable or the app is clunky, people will just ditch it, right? No matter how good the science is.
Absolutely. Patient input makes it more likely they'll actually use it consistently. It reduces technical dropouts, people giving up due to frustration. Better adherence means less missing data.
And for the listener, this means future health tech should be designed better for practical use, not just clinical accuracy.
Exactly. And finally, there's the power of watching things in real time. Monitoring dashboards.
Vigilance is key, you said.
It is. Using dashboards to track compliance in real time is incredibly powerful. Researchers can see instantly who's wearing their device, how consistently, if data is flowing properly.
So they're not waiting weeks to find out there's a problem. They know right away.
Exactly.
Yep.
Which enables proactive intervention. Dashboards can trigger alerts, low wear time, maybe a device failure signal.
Allowing researchers to jump in quickly.
Yes, they can reach out, remind someone, troubleshoot a technical issue, offer support before a huge amount of data is lost. Imagine a dashboard flag popping up if someone hasn't synced data in 12 hours. A quick call could solve the problem.
That proactive engagement can save so much valuable data. It's an adaptive approach.
It really is. But even with the best prevention and imputation, there's a final crucial step: ensuring robustness. Rigorous statistical modeling and sensitivity analysis.
This is where they really stress test the findings.
Absolutely. Before drawing conclusions, you have to test assumptions about why the data is missing. Is it missing completely at random? MDIR. Just totally random.
Or missing at random, MAR, where missingness is related to other observed data.
Right. Or is it that tricky, not missing at random, NMAR, where it's related to the missing value itself?
And why is knowing that distinction so critical?
Because the statistical method you use to handle the missing data must match that underlying mechanism. Using an MDR method for NMAR data, that leads to biased, wrong conclusions.
Which is why you need sensitivity analyses.
Mandatory, really. These analyses explore how stable, how robust the study conclusions are under different assumptions about that missing data. You rerun the analysis multiple times using different methods or different plausible scenarios for the missingness.
Trying to see if the main conclusion still holds up, no matter how you slice the missing data problem.
Exactly. The paper mentions powerful examples like worst-case imputations.
That sounds intense. What does it involve?
Imagine your new drug is supposed to increase activity. Worst-case imputation means you reanalyze, assuming every single missing activity data point represents the worst possible outcome, like zero activity.
Wow. So even if data is missing, you assume the participant was doing the worst thing possible during that time.
Yes. It provides a very conservative lower bound for the treatment effect. If the drug still shows a significant benefit even under this extreme pessimistic assumption, well, that builds incredible confidence.
That's a seriously tough test. If it passes that, it must be pretty solid.
It's very stringent. Another technique is pattern mixture models. These explicitly model different groups based on their missing data patterns, like analyzing dropouts separately from completers, acknowledging their missingness might be different and linked to outcomes.
So you're directly accounting for those different patterns.
Yes. By running these what-if scenarios, these rigorous challenges, researchers demonstrate their findings are reliable, consistent, not just an artifact of how they handled missing data.
It's about building multiple lines of evidence, building trust.
Exactly, enhancing inferential robustness. And beyond sensitivity analyses, specific models like mixed effects models are really useful here too.
Why are they good for this?
They're great at handling unbalanced data common in longitudinal studies with missing bits. They can model changes within each person over time while also accounting for differences between people all in one go.
So they handle individual journeys and group trends simultaneously, even with gaps.
Yes, very powerful. And finally, there's adjusting for missingness within the formal estimand framework. This aligns with major international guidelines like ICH E9R1.
Demand framework. What does that do?
It forces researchers to be incredibly precise right from the start about what they're trying to estimate in the trial, and crucially, how they will handle intercurrent events, real-world things like missing data, people stopping treatment, using other meds.
So you have to declare upfront how you'll deal with all that real-world messiness, like writing the rules before the game starts.
Precisely. By defining and adjusting for missingness within this framework, the analysis perfectly matches the scientific question and meets regulatory expectations. It leads to clear, unbiased conclusions.
And for the listener, all this complex stats work means you can have more confidence that the conclusions about a new drug are solid, rigorously tested, and transparently reported.
Absolutely. Which brings us neatly to the regulatory side, because all this work ultimately has to satisfy bodies like the FDA.
Right, they have the final say. And they care a lot about missing data.
Immensely. It's a major focus. The source paper highlights the FDA emphasizes pre-specifying missing data handling in analysis plans. You must outline your strategy before the trial starts. No making it up later.
You can't move the goalposts. It's about transparency and preventing bias.
Fundamental for integrity. And the consequences are clear. The paper states, unmanaged missingness introduces bias and may lead to regulatory rejection of digital endpoint claims.
So, even if a drug looks good initially, if the missing data wasn't handled properly according to a preset plan, the whole thing could be rejected.
Correct. It's a critical patient protection measure. Ensures only rigorously validated digital endpoints support new therapies. The paper's authors argue that structured, documented missing data strategies are now core to trial readiness and data integrity. Not just good practice, but essential for success.
Okay, so let's pull it all together. What are the absolute key takeaways for you, our listener, from this deep dive?
First, imputation isn't optional anymore. It's essential. Using techniques like within-patient imputation, FDA, multiple imputation, even deep learning. These are crucial tools to get robust, valid endpoints from DHT data. We can't just ignore gaps or use simplistic fixes.
Got it. Imputation is key. What else?
Second, proactive mitigation is incredibly powerful, maybe the most powerful strategy. Preventing data loss through smart design, good participant training, real-time monitoring. It reduces the problem significantly from the start. Build a solid foundation.
Prevention first. Makes sense. Third,
sensitivity analysis validates your findings. You must rigorously test how robust your conclusions are under different missing data assumptions. It builds trust and credibility like nothing else.
stress test those results. And finally,
Documentation and alignment are non-negotiable. You have to thoroughly document every missing data strategy in the protocol, the analysis plan, ensuring it aligns with regulatory expectations. This guarantees the endpoints are valid, defensible, acceptable. Transparency is paramount.
so imputation, prevention, sensitivity analysis, and documentation, that covers a lot of ground.
It does. And all this complex, often unseen work, is what ensures the data-driving future health decisions is truly sound.
We've definitely taken a comprehensive look today into this, well, surprisingly complex but absolutely critical world of missing data and digital health trials. From the challenges of high-frequency data to cutting-edge AI solutions to proactive strategies, we've seen how much goes into making sure that data is reliable.
And that behind-the-scenes rigor is what ensures the digital health tools, the treatments tomorrow, are built on solid, unbiased science. It's about confidence. Confidence in the insights from your own data and confidence in the medicines that come to market.
So here's a final thought for you to chew on. As these digital health technologies become even more embedded in our lives, tracking sleep, steps, heart rate, maybe even mood, how might our own habits, our consistency or lack thereof in using these devices, how might that impact the precision and reliability of future medical discoveries that really depend on that continuous data stream?
It's a fascinating question, a responsibility perhaps we all increasingly share.
Definitely something to think about. Until next time on The Deep Dive, keep exploring, keep learning, and keep asking those deep questions.